positive blood group or a negative blood group. Why do we say some people have positive, like I'm B positive? If someone is O negative, why that person is O negative? No idea? Okay, so there is another type of protein present on our like genes, which is giving us a positive or a negative value that was first discovered in the rhesus monkey, and it is named as rhesus. It's a rhesus factor. If it is present, then it would be positive. If it's not present, then it would be negative. What's your blood group, by the way? O negative. It means you do not have A antigen, you don't have B antigen, moreover, you don't have the rhesus factor. Okay, everything is absent in you. Okay, that's why O negative is like a universal donor, because whenever we give such blood group to someone, because nothing is present, it has, there would be won't be any agglutination when it is transferred to some other person. Now what is agglutination? Means if there are coinciding genes, means if you have A and some other person have B, and you are transferring your blood group to that person, your blood will clot. That's why we cannot give the opposite, like, or different, the complementary blood groups. We need to stick to the same one. So O is considered as a universal donor because it has nothing, like nothing, so there won't be any agglutination. Okay, they have given you a key for what is A, what is ABO, and everything. Okay, now when they mention A, there can be two cases. It can be both A, as they have given. It can be both A, or it can be both, like IA or IA, or it can be IA, sorry, or IO. Do you know why we write this word I for blood groups and not for others? Okay, so we say this is one immunoglobulin protein on which these antigens are present, like A or B, which actually determines our blood group. That's why we write it with I. Okay, give the genotype of ABO blood group for individuals 1 and 2. So if you look at this key, what is your like conclusion? What would it be? IA, IB, what? The Okay, A would, we have to give 1 and 2. Okay, 1 would be A. So we can write, I've given you a case for A. So we have two cases of A. Either both can be dominant, like capital A, capital A, or maybe one of the immunoglobulin is absent. Okay, so we can say it can be I, A, I, O. And in case of AB, both alleles would be present. It would be I, A, and I, B, because both are present. Okay, next one. The next statement is explain one piece of evidence from the figure above that the alleles for rhesus positive is dominant. Let me take the screenshot. Here we have again the calculation. Okay, so what's your answer? Yeah, B. Like what information are we getting from this, like this pedigree? So here we have a negative one as well. So if we have a cross of 3 and 4, it's evident that one of the offspring has a like a negative means that recessive allele is or is absent. Rhesus allele is absent. Wait, wait. Okay, look at this pedigree 3 and 4. I thought you're not getting it. Okay, look at 3 and 4. Okay, And then have a look at 5, 6, 7, 8. These are offsprings of 3 and 4. Okay. Like one is, sorry, the second one is no, it's no, it won't be carrier. Like it's absent. It won't be carrier. Like absent. We need to mention it absent. Okay, wait. I'll let you know why is so. Or for example, if it has a positive, or the recess is present positive. Okay. Now, when we make a cross of it, it means the organisms are going to have this positive factor. Even though if it is absent in this one, this A is going to transfer this positive state character, or you can say the state, to the next one. It means that this allele is dominant. Now this is fourth one is not an offspring. This 3 is married to someone with 4, with again a positive allele. Now here we have chances. Here we have chances that there is positive and there would be no, it's not present. There are two chances. So when it is transferred to the next, like offspring or next generation, this again, if it have both are absent, like maybe this female or male would also not have this one. O. It means both are getting O, so this would be negative. Okay, this is a pedigree. We need to find the chances. Now we have to give one evidence that this allele is recess allele is dominant, or positive is dominant, because it is transferring in most of the cases. Like here, it's transferring, and here it's a 75% of probability that they would have a positive blood group. That means it's making it dominant. Okay. Next one. Explain one evidence from the figure above that the this we have done already, sorry. Calculate the probability of 1 and 2 producing rhesus positive son with the blood group A. You can assume that individual 1 is heterozygous for the rhesus blood group. Okay, let's see this one. uh Wait, let me open it on another screen for me, for myself. Okay, we have a blood group A in 1. So 1 is heterozygous. So it is IA and IO. Is it so? Moreover, it is positive. Let's place a sign plus on it. Okay. Now we have to cross it with B, and this is also heterozygous with 1A and 1, sorry, IA and IB, and nothing is present. Like no rhesus protein is present. Have a cross of this one. They have a positive, okay, they are saying they have a positive blood group A individual 3. Okay, have a cross of this, and then find the probability. Like first of all, make a cross, yeah, and then find the probability. And IO positive. Okay, this would be IA and IB. No positive sign would be there. Okay, now write it. Is it half? 1 by 2? 1 by 8. Okay. How have you calculated 1 by 8? It's 1 by 8, but how have you figured it out that it's 1 by 8? Okay, wait. Let me. Have you figured it out that what it is? Okay, okay. Let me. First of all, we'll write the answer. What is the answer for this one? IA positive with IA, and then we have IO with IA, okay? IA positive with IB, and then we have IO with IB. Okay, then we have two positive persons and two negative persons. Is it so? Now let's calculate how many are males and how many are females. So it's a probability that one would be male and one would be female. Okay? Now there is a probability formula that we write for it. Okay? So we have to consider all three factors. We have to tell for male as well. We have to tell for uh, the blood group as well. It has a blood group A, is it so? Okay, here it's mentioned. We have to mention the blood group. Oh, sorry. And the third factor that we need to keep in mind, though we multiplied, third factor that they're mentioning is the RH factor. Okay, these three factors we are considering. Now we say half of the offsprings are male. Is it so? Half of the offspring among these, we are talking about these. Half of the offspring have A blood group. Okay, half A, half male, and then half of them, like out of these four, half of them are positive. So half are positive. Okay, now we need to multiply it. Multiply it, it would become 1 by 8. This is your probability. So what would be your answer if we write it in percentage or the other value? It would be 0.125, okay, or 12.5%. Have you got it now how to calculate it? Okay. So whenever they ask you to find the probability, you first of need to write the phenotype. After writing the phenotype, you need to calculate what have they asked for. They have asked us for third individual. You have to check, okay, if this is the first a third individual and this has this one characteristic, it is positive, it is male, and it is like have a blood group A. These are the three characteristics that they have explained in the table. So you need to mention these characteristics. If there are more, then we add 1 by 2, 1 by 2, 1 by 2 more. Okay, I hope we are getting more questions like this in the other like questions as well, so that it can be practiced. Okay, again we have a Hardy Weinberg equation, and here we have to find the frequency of IB. Getting me? Not getting me? Okay, wait. We have three alleles. We have IA, we have IB, and we have IO. Are these are the three alleles we do have? So when we say the frequency is always 100%. That is 1. 100% means 1. Okay. So let's say 1 is equal to, let's say, what is this IA? This IA is 0.15. This IO is 0.65. This IB is missing, which we need to find. So 1 is equal to what would be addition of this? This one be 0.80. Okay. 1, now if we want to calculate IB, we need to take this 0.80 on the other side of equal. It would become 1 minus 0.80 is equal to IB. So what would be answer? It would be zero point 0.2. Done? Or you can also write 0.20 because it's 80, then it would be 20. 0.2 is your answer. I hope you got it. We need to read the statement. I misread the earlier statements. This the other question statement. We need to focus on the statement. What statement is being asked? Sometimes we do not need to apply the whole Hardy-Weinberg equation. We just need to find the answer. Okay, if this is the case, this is the case, what could be written? Okay? Okay, thank you so much. And we'll definitely share a paper with me uh, before class so that I can like have a review of it, just go through the questions because sometimes when I'm reading like for the first time, I need to like understand the whole equation, what is being asked in question. Okay? Sing like you. We need just need to go through the like statements, what statement is given. Okay, thank you so much. Have a great day. Goodbye.